Many thanks, Glyn, for inviting me, and thanks for the audience for coming. So I would try to explain why the Human Embryonic Stem Cell Registry, as it was called when it was implemented in 2007, and now the Human Pluripotent Stem Cell Registry was implemented, and how the registry qualifies data on these cells. So first I want to briefly talk about why we want to qualify pluripotent stem cells, and then how we do that with ethical aspects on how these cells are being derived and the tissue from which these are derived are being collected, and how we try to qualify the scientific criteria to say that it's a pluripotent stem cell in this area. And finally, I want to show you briefly how we implement it in hPSCreg. So this graph shows the development of research paper output using embryonic stem cells or induced pluripotent stem cells until 2013 based on PubMed and other public resources. And the 2014 data would show you that both cell types will have parallel increase in paper output. So there is a large number of research, large amount of research being done with human iPS cells as well as with human embryonic stem cells. And one of the reasons to implement a qualifying procedure for these cells, for European research at least, but also for global research, was that we want to know what kind of cells are being used. Are these cells do these cells adhere to certain standards? We want to see that, and we want to also make the research comparable to these standards. So that was basically the origin of the Human Embryonic Stem Cell Registry. We said, okay, if you want to control or at least have an overview of what kind of cells are being used, we need to register these cells. And this was especially important at that time for human embryonic stem cells, not only because of the ethical debate, but also because we wanted to make sure that the cells which are being used are adherent to certain quality standards. So there are 520 papers on embryonic stem cells and 468 papers published on human iPS cells in 2013 and 14, as I said, will be much more. So the question was then, so how we qualify it and what do we qualify? The first issue is, are these cells really pluripotent? And the second question was then, how these cells have been collected. So where are the ethical standards for their collection? Or what kind of ethical standards have been applied? And third, how is that validated? Right? And we can see here that the risks which would pertain in the field, which would be in the field if there is no qualification, central kind of registry. And of course, there would be no peer review for many of the existing lines. The published lines are less critical, but we will see that there are very few lines. And lines would potentially be used without any quality check. This can also lead to contamination of purely characterized cell lines, and these contaminations in results and of cell lines are then inherited in the data resources. And you would also have difficulty in locating certain cell lines if there is no central registry. You want to work with a certain qualified cell line, and you're not sure whether the lines you access are adherent to these to your quality criteria. So first I want to talk a little bit about ethical qualifications. So what kind of ethical criteria do we put on a cell line to say, well, it is qualified to be used in European research? And there are several aspects to it, and one is, of course, the main point is consent, is there informed consent, but there are also other issues like data and how the samples are stored and accessed, for example. And we have tried to structure that a little bit. And the most important thing we asked when we get a cell line to be registered, is there consent available? has a donor expressed what a researcher can do with the cell line. And this is then the basis for all other things that people can do with a line. So consent, the donor is informed what is being done with the line, but the donor has also to say, what can you do with my line? And this is not only relevant for the tissue and for the lines derived from this tissue, but also for the data associated with these tissues and cell lines, and also with the donor himself or herself. So how are the data handled? How is privacy protected? And how is the misuse of these data prevented? So as I already said, the donor needs to permit the possible use of the lines or of the tissue for the research. And also this implies not only the research, but also later application, commercialization. So it must be clear that the donor is aware of these kind of later applications of the lines. And finally, although it's not strictly as an ethical aspect, but it is relevant for the user of the lines and also eventually for the donor, what are the regulations and the rules that need to be applied to use the tissue or the cell lines. For example, are there data protection rules which are applicable, and are these rules implemented? So in ethical can be seen as ethics in general. The ethics background of a cell or of tissue can be seen as an idea, as we heard in the previous presentation, but it can also be structured. And we try to codify or structure an ethics consent or ethics criteria which are necessary to qualify a cell line into a kind of table. And we asked everybody who wants to register a cell in the registry certain questions. Some of these are absolutely necessary to be answered, and some are just additional information, which might be relevant for some people who want to apply the cell line, for example, commercially, but not really relevant for the ethical procurement of these cells. So the first question we ask, of course, is, has consent been obtained from the donor of the tissue from which the iPS cells or the embryonic stem cells have been made? And was it consent voluntary? Was there any pressure put on the donor to donate the tissue? And can we have a copy of that consent, please? So that's the first thing we absolutely require to make a cell line registered or accessible. If you don't have the donor consent, who has it? Can we get it somehow? Right, and what is the content of that information, of the consent information? So this is the first thing we want to know. And does a donor know how the data are protected? And is it pseudonymized? Is it anonymized? Is it traceable or not? Because that has also implementation on how we handle the data we get about the cell and the scientific data or the donor information, the medical records. And has the donor been informed whether the donation of the material impacts in any way the personal treatment? So the second aspect we ask is permitted usage. So users want to know when they go to a registry or when they have a cell line, what can I do with it? Can I only use it for academic research, or can I also use it for clinical application, for example? Or is it just restricted? Is the use restricted to a very specific research topic? So, which prohibits any use of the cells or of the data associated with the cells for future research. So we asked for what is the permission, how far is the permission going, and is it for a research organization, is it for commercial application as well? Can it be used in a company or only in a non-for-profit company? These are not strictly, we don't ask, this is not a mandatory information we asked, but it's a very interesting information for later applications, and specifically it's an interesting information for cell banks who want to sell these cells for example, to pharmaceutical companies. If this condition is not there, pharmaceutical companies are probably not interested in using these cell lines. And this is the next topic I put on here, permitted use is commercial exploitation. And one issue which I highlighted here a little bit is financial gain permitted when using my donated tissue or the cells derived from it? Can the company make money from it, or is it explicitly prohibited? We have here, you might have noticed that we have two kind of wording here, two kinds of wording. One is permit. Is it permitted explicitly, or does the consent prevent the financial gain? So this is an issue in discussion. The permission is explicit, but preventing does only mean, well, has it been explicitly said, no, please don't do it? If not, there's still an open question, can it be done or not? So that's very often this is not in the consent information of existing lines, so we asked for it, and I don't know exactly how legally binding that would be. If you say here, it doesn't prevent financial gain. So then there is a question how we store the material, how long are we allowed to store it, and can the material be available anywhere in the world? So is it restricted to one country, to one institution, or only to Europe? So what are the benefits for the donor? Will the donor expect some financial benefit when he donates tissue? Right? Directly, because he is donating something from his body and he wants to get paid for it. This is not allowed in the European directive, but it can happen. And what is allowed is that you get reasonable expenses reimbursed. So this is not meant here. It's really meant a payment for donation. And another benefit would be if there is a commercial product which generates profits, is a donor entitled to a share of that profit? This is also something very interesting for pharmaceutical companies, for example, who want to use a cell line and eventually make a profit from it or from the results of their research. So they will probably not use a line when there is some benefit in terms of profit sharing to the donor. So what about data? We have heard about data in the previous talk already, and this has something to do with the first question we asked, is the donor anonymized or is it pseudonymized? So that somehow is associated with how the data can be accessed or accessed. But this is not enough. Even anonymized data, if it's genetic data, can be used to re-identify a donor. So privacy protection is not guaranteed by just anonymizing the data with a code. It needs more protection. And so we asked whether the donor want to have the data absolutely freely accessible by anybody. So then it's an open access procedure, or managed. The access is managed by a data access committee. So a second instance which then permits or doesn't permit the use of the data for certain research purposes. How is incidental finding handled? So many cell lines are tested for certain pathogens like HIV, HPV, or HCV. Some of these need to be relayed back to the donor because it has a severe health impact, and the donor needs to be informed on how that is being handled. Does he want to get the feedback, or does he not want to get the feedback? In some countries, the law requires feedback, for example, in the case of HIV infection, identified HIV infection. So medical records has a similar data protection issues as genetic information. So can we have access to medical records about the patients, and how are these medical records being protected? So we have seen that. I will not go into that. So in the European Union, there's an ex-ante review process. We have heard about why that is, and that it protects the researcher, basically from later lawsuits. And the registry is part of that process. So we, as a registry, evaluate on the ethical level the cell lines which are being used in the European Commission, in the European Union, for at least for European-funded research. So we fill out certificates and say, yes, in our opinion, as far as we can validate, this cell line fulfills the ethical standards needed to do research with a line in Europe. The other issue is scientific qualification. So if the ethical standards are okay, then we ask, well, is the cell really pluripotent? This is the first and most important scientific qualification criteria for us, how we assess that. And we have developed two basic criteria. One is phenotype. So are there markers expressed which are typically expressed by pluripotent stem cells? And is the morphology of the cell similar to a typical pluripotent stem cell line? And there's functional evidence. So can the function of a pluripotent cell is to differentiate, at least by definition, in cell types of all three germ layers. So can the cells do that? Is there evidence to show that they differentiate appropriately? So we ask that information. And we also want to know what is the disease background of the donor of the cell line. So this is, in my opinion, an important scientific information about the cell line. And we try to collect this data as well. So how do we do the scientific qualification? It's shown we asked, again, as in ethics, we have a questionnaire. And we asked, well, provide the data, please, which you have collected to show that these are pluripotent stem cells. And the first is provide the markers, the pluripotency markers you have detected to be expressed in your cell lines. And you can get a window, and then you can use data which are already provided in a list or add new data to it, new information to it, and then we evaluate that and say, okay, when at least four common pluripotency markers are expressed, then these criteria is acceptable. And there's more information, and I already mentioned the disease. So does the donor have a disease, or is there no disease? Another information which we think is important is the traceability of a line to a certain donor. A donor, first of all, you can derive several cell lines from a donor. We have heard StemBank derives 3 cell lines from 3 clones, so to say, from one donor. And a donor can switch from one institution to the next and again provide cells, and another cell line is derived. We want to know which cells which are around come from the same donor. So there need to be some way of traceability back to one donor of many different cell lines, perhaps. And we also want to know where the cell line comes from which kind of institution. To trace a donor, we have developed this standard name or nomenclature which codifies this information. So codifies every donor of a cell line in the first 3 blocks. Basically creates a unique donor identifier, which is composed of an element which has a name, the institutional acronym, identifies whether it's, for example, an iPS line or an embryonic stem cell line, and has an additional 3 character alphanumerical code, which allows basically each institution to at least derive more than 40,000 or cells from at least 40,000 donors, which we think is, for now, it's enough. But then we also have a clone identifier. So clone number 1, 2, and 3 of one donor would be ABC, clone ABC, and we have even subclones because some of the cell lines will be modified, genetically modified to make, for example, isogenic clones, repairing a genetic defect or setting out genetic defects. That would constitute subclones, and we can associate these subclones as well to a parental cell line and also to the donor. So we we thought this traceability is a relevant, very relevant aspect for qualifying a cell line, and we implement that now in the registry to be sure that there's no mix-up of cell lines. So I want to go a little bit back. So these are the mandatory qualifying scientific criteria. So is there evidence of pluripotency shown by marker expression, by morphology, and by differentiation potency? And what is a donor identity? So we assign a unique donor identifier to it. So, but for researchers, this is often not enough. They want to have evidence that the cell will work in their experiments, and they don't want to have failures. These are expensive experiments. Failures are prohibited. So many researchers use established lines, and they use these lines all over the place, all over the time, because these have been proven to do certain things well. And this is a list we compiled also in 2013, and this is what kind of lines are being published, are used in published research. And you can see that the field is completely dominated in the embryonic stem cell area, that is, by two lines. This is H9 and H1 from Y cell. Then the next place is again Y cell line H7. There's another Y cell line H14. And there are a few other lines. And most of the lines are from the US. Some lines are from other institutions, Kyoto or ES Cell International. But it's completely dominated by a few lines. And the same case could be found for iPS cell lines, although this is a little bit more diverse, as people increasingly mix their own lines or use lines which are deposited in many, many different banks. But also here, for iPS cell lines, if you trace back the iPS cell line to a donor, you will find that many people used fibroblasts from Coriell or from ATCC. So it's basically one donor, which is being used for many, many cell lines in many, many labs. This is also a challenge for the nomenclature and for the traceability, of course, but it also shows that actually you think you use many different lines, but actually use only line from one donor. So how can we change that using hPSCreg? And we thought we can only change it when we provide as much information on every line we have registered as possible. So we have tools, we have more than 1,000 fields actually, which I don't want to go into detail here, to add additional scientific information, which is not mandatory, but is very helpful to make the line much more usable for the community. And one is, for example, karyotype of the line at different passages, or genotype of the line, using different methods. The genotype we are again run in data protection issues, because the genotype is deposited not in the registry, but in other databases, and the access to these databases is controlled by a data access committee, for example. And but it is, we know the information is there, and if somebody is interested in that, it's possible to get it. So we have, as I said, more than 1,000 fields which can be filled out to get additional information on the cell line. This is actually how when you go to the registry, this is a front page, you can search for cell lines and you can register a line, but you can also browse the registry for certain information, for example, for research projects or for diseases. So lines with certain diseases, you get this information. And this is research projects. We thought one important information which was relevant for the widespread use of H1 and H9 in the ES field was social communication. So did you use the line? Yes, I used it. It worked. I will use it as well. So if we provide the information who in Europe uses certain cell lines in their projects, it adds confidence in the workability with these lines. So we, at least for the European funded projects, we have most projects in the registry and we know which lines are being used there. So we hope to promote communication between the projects and make more European lines used in Europe. So US lines. There are also IP issues, which I don't touch here today. So how then we validate all these data? We have a process in place, which is very briefly here. So minimum data are submitted with the cell line. The cell line gets a standard nomenclature name, and then the data will be registered and validated. This is, well, anyway. Then they will be validated by a two-tiered process. So we validate it within the hPSCreg team, and we, if there are any questions, we go back to the researcher or to specialists in the field to provide support in validation. Then the cell is only after validation, the information is released. The data can be taken out from the registry by an export API, application program interface, and you can add more data at later stages to each registered cell line. This is a function which is not yet implemented, but we think it's very important. So to get as much data for each cell line is not easy, but it can be done basically in two ways. One way is we collect all the data and deposit it in the registries themselves using a huge server. This is not the way we want to do it. We want to access other databases which have data. At least we don't want to do it for all data types, but usually we want to access other databases which have these data. One example was already mentioned, is the genetic data for cell lines, which are in, for example, in EBI or NCBI. Another tool we have already established is called CellFinder, and in CellFinder we use an ontology. Who was here yesterday knows that also in ToxBank, and Barry was talking about these structured nomenclatures of terms which can can codify any type of data basically, and this will then be used to organize all different types of data in a database. In this case, it's called CellFinder, and we want to use CellFinder to get a lot of data associated with the cell lines in the registry. Pictures, for example, or gene expression data, or PubMed IDs for publications, and graphs, many other data. Connect these data via the unique identifier or ontology identifier, and have them search them in CellFinder and in hPSCreg using these identifiers. So CellFinder looks like that. You can use it for data integration. When you go there, you can have all types of cell types registered there in the CellFinder database, and you can search for it, and then you can connect with embryonic or pluripotent stem cell line in hPSCreg in the registry, and ask, well, has this line been differentiated, for example, in any cell type, which I also find in CellFinder? And is this cell type a mature hepatocyte or a fetal characteristic of a hepatocyte? So this is the basic idea. So I will only go into one of the three major search tools we have. So this is a so-called buddy browser. We can, of course, also analyze expression data and compare cells based on the expression data, or we can see where in the developmental tree from a zygote to a mature organ the cell I have in hand belongs to. Where is it positioned there? I don't want to talk about that. I just talk about the buddy browser. So what we can do in CellFinder in theory is have a pluripotent cell and have every cell in between this cell and the mature terminated differentiated cell characterized with all the data we have in the ontology identifiers. Practically, you can basically either go through this developmental tree, which is schematically here, or you just go, well, I want to see whether my cell I have is really a kidney cell, for example, and I can search for this identifier in the body, the human body, go to the kidney, and see all kinds of information which I have in the database on this organ, and I can zoom into it in the kidney and get more details about the organ. For example, go down to the glomerulus, and I'm interested in this podocyte, for example. I can see images of this and get more information on the right side, principally. So I can zoom in from kidney to the nephron structure, to the glomerulus, to the single cell, and for each of these stages I get information about the cell. And when I have differentiated a cell from a pluripotent stem cell, I can compare the information with that. We can also do that on the gene expression level. I can basically go to an organ and ask, well, is my gene expressed there, and where else is it expressed? So is it a marker for my organ, or is it also in other organs expressed, and to what level? This uses data which are, again, in the World Wide Web accessible in many, many databases collected here, compounded, and then you can analyze your body or your cell type. It doesn't need to be on this level. It can be on any of the zoom levels I have shown. So in summary, I just want to say, well, the registry is should be a one-stop information hub about quality-controlled human pluripotent stem cell lines. There's an ethical validation of the ethical procurement of the lines. There's a scientific quality control of the lines registered. And only after that is satisfactory, we will release the cells to the public and make them visible to anybody, and allow access and comparison between the different cells and their information. What is not yet implemented, but what I think is very important, I mentioned it before, is the feedback, integrate user feedback. So what actually, if I use the cell lines and it didn't work, or it did work in my application, I want the feedback in the registry. So there is a body on information built up on every cell line being used, which is interesting for other research to make a decision whether the cell lines is also useful for their particular experiment. And with that, I want to thank you.